Hi, my name is Robert Jacobs, and I'm an assistant professor at Western Carolina University in the Department of Health and Physical Education and the Department of Physical Therapy. The Physiological Society generously supported a symposium entitled Skeletal Muscle Adaptations to Endurance Training. Is Tissue Hypoxia the Main Signal?, which was held at this year's annual Congress of the European College of Sports Sciences in Malmö, Sweden. The main reason for organizing the symposium was to discuss the influence of hypoxia on the response of skeletal muscle mitochondrial adaptations to exercise training, since exercise-induced tissue hypoxia in general is expected to facilitate mitochondrial biogenesis. In contrast hereto, hypoxia as experienced with exposure to high altitude has been demonstrated to decrease skeletal muscle mitochondrial properties. To discuss these apparently conflicting traits of hypoxia, experts within the field presented their views, and the symposium reviews included in this issue of Experimental Physiology summarize these. Research from the lab of Carsten Lundby first discussed basic adaptations of skeletal muscle mitochondria to classic endurance exercise training. While mitochondrial volume density increases with most endurance exercise training modalities, the specific alterations in the electron transport system appear dependent more on the intensity of exercise. The capacity for fat-stimulated respiration, or maximal electron flow through electron-transferring flavoprotein, seems to improve the most with all types of exercise training, and this improvement, which is about 25% increase, is independent of training intensity. It makes intuitive sense that this characteristic is improved the most, as superior whole body fat oxidation rates are a key feature of athletic populations. Since it has previously been shown that skeletal muscle mitochondrial oxidative capacity exceeds that of systemic oxygen delivery, it also makes sense that increases in oxidative phosphorylation capacity are not as robust as the increases in the capacity to oxidize fat. And perhaps the changes in oxidative phosphorylation capacity with endurance training may simply be a consequence of an overall training-induced mitochondrial biogenesis and not specifically aimed to increase its maximal rate. Recent data from a study published from Dr. Lundby's lab suggests the role of mitochondrial adaptations to classic endurance type of training for improvements in VO2 max seems limited, but has relevance for submaximal type of exercise. In the review by Dr. Andrew Murray from Cambridge University, he discusses the effects of high-altitude exposure on skeletal and heart muscle and explains how in humans returning from altitude or following short-term hypoxia in a chamber, cardiac muscle energetics are altered, suggesting a loss of energetic reserve. While the underlying mechanisms remain unresolved, in hypoxic rats, mitochondrial function is altered with a particular loss of fatty acid oxidation capacity and downregulation of genes associated with fatty acid oxidation. In skeletal muscle, however, Dr. Murray argues that the picture is less clear. In climbers returning from extreme high altitude, there is a loss of skeletal muscle mitochondrial density, though interestingly, no such change was seen in subjects following a simulated ascent of Everest in a chamber. Mitochondrial function appears to be altered following longer-term exposures to high altitude, with a loss of respiratory capacity but improved mitochondrial coupling, suggesting improved efficiency. There's also a possible loss of fatty acid oxidation, though this is not a consistent finding. Intriguingly, though, despite such changes, muscle capacity for ATP synthesis following an exercise challenge appears to be relatively well preserved in individuals returning from altitude. Dr. Rundqvist highlights that in addition to systemic hypoxia experienced at high altitude, local oxygen levels also drop in skeletal muscle during exercise due to increased oxygen demands. Cellular response to hypoxia is to a large extent conferred by activation of the hypoxia-sensitive transcription factor, HIF1, and skeletal muscle levels of HIF1 have been shown to increase in response to acute exercise. The target genes of HIF1 increase oxygen transport through, for example, EPO-mediated erythropoiesis and VEGF-induced angiogenesis, and/or improve tissue function at low oxygen availability through increased expression of glucose transporters and glycolytic enzymes. However, HIF1 may also inhibit cellular oxygen consumption and mitochondrial oxidative metabolism, both features discordant with the phenotype of a trained muscle. Dr. Rundqvist emphasizes that the acute activation of HIF1 during exercise is blunted after a period of endurance training, possibly through a high expression of its negative regulators of the prolyl hydroxylase family. This long-term adaptation may be necessary to fully enable the enhanced oxidative metabolism that is part of a local physiological response to endurance training. Dr. Murray also stresses that although chronic hyperbaric hypoxia is the defining feature of the high-altitude environment, and as such oxygen delivery to the tissues is challenged, that other potential confounding factors, such as changes in temperature, absolute humidity, radiation exposure, challenges to the diet, and possible high-altitude related illnesses make high altitude an imperfect setting in which to investigate the physiological response to hypoxia alone. But it is nevertheless a worthwhile research endeavor due to the number of people venturing into high altitude regions for work or leisure each year. Since the publication of Dr. Murray's review, recent research published from Dr. Lundby's lab has demonstrated that in humans exposed to hyperbaric hypoxia, but in otherwise controlled conditions, mitochondrial volume density increased. This may suggest that at least some of the previous observed differences between alterations in the mitochondrial profile following exercise training and altitude exposure may not be as substantial as previously thought.